Today we will discuss the genome, that is the genes and the chromosomes. As you know, in a living system, the chemical basis of life is the DNA molecule. The DNA, that is the deoxyribonucleic acids, contain the nucleotides. This is the monomeric units of the DNA, nucleotide, that is the base, sugar and the phosphate. So, the triplet code of this genes or which is present in the DNA, that the segment of these nucleotides which are known as the genes, and the collectively the genes are known as the genomes. So, we contain near about the DNA contain near about 20 to 23,000 of the genes. That means every cell or every living system, the genes which are present or functionally we expect that near about 20 to 23,000 of genes, and previously we were thinking that near about 30 to 40,000 genes are there, but gradually when we saw that this number is little bit less, that is the 20 to 23,000 of genes are there, and the genes are they expressed, and that is the central dogma is the DNA to the mRNA, that is the transcription, and the protein synthesis known as the translation. So, that the presentation I have already taken from the Google's, the presentation slide have been, some slide has been taken. The genetics, as I was telling about that the heredity of the life, that it passed from the information, that is the chemical information from generation to generate. Genetics is derived from the Greek word generate, to generate. So, genetic is a branch where we usually study about the heredity, the inheritance or the inheritance pattern, that is the science usually deals with the study of the inheritance. The chromosome is which is present in the nucleus. This the DNA contain near about 600 crores of the nucleotides. So, this huge number of nucleotides in the DNA, that is packaged in a organized fashion, because in the nature the cell contains so many organelles. These organelles are the organized structure, it is not haphazardly present. So, genetics material, this DNA is packed inside the cell, that is in the nucleus, in a specific manner. So, the DNA is the deoxyribonucleic acids, it is packed. So, packaging is very systematic phenomena by the nature inside the cell, that the chromosome, the DNA is packed to a chromosome, that is the DNA double helical structure is 2 nanometer diameter structure is packed to the chromosome, which is a 1400 nanometer structure. So, initially what happened, that DNA molecule wrap around the protein, that is the histone protein, which are basic protein. We contain near about 5 type of histone, that H1, H2A, H2B, H3 and 4. These histone proteins help as a platform where this DNA molecule wrapped, that one nucleosome, that is the one basic unit of the chromosome, that is DNA wrapped into this histone protein, this core protein, that it is a octamer 2 each, that is 2A, 2B, 3 and 4, 2 each, that remain in a one core, and H, that is the histone 1, H1 remain in the periphery. Just assume that is a knot, put a knot to fix a knot, that H1, that histone protein acts as a stabilizing the knot. So, this DNA wrapped around this protein, basic protein molecule, the histone protein, that near about 146 nucleotide base. It is the 1.75 tons, like this, it is wrap around, that is the 1.75 tons, and that contain near about 146 nucleotides, and the flanking zone it contain near about 60 nucleotides. So, it almost pegged to a one nucleosomes, that is the one unit, and the multiple nucleogen is known as chromatin, and then the this chromatin wrapped and like a solenoid structure. So, this 2 nanometer structure to one nucleosome, it is 10 nanometer structure, and this 10 nanometer structure again wrap around in the like a solenoid, that it is 30 nanometer. This again it not, it is pegged to 300 nanometer, again then 300 nanometer pegged to 700 nanometers, and again it is pegged to the 1400 nanometer. So, one unit of chromosomes, that is pegged, this DNA pegged to the 14 nanometer structure. So, 2 nanometer structure pegged to the 14 nanometer structure. In the human being, this number of chromosome varies. This chromosome, it is vary to specific species to species. Human being, there is 46 number of chromosome, that is the 23 pairs of the chromosome human being contain, that is the human cell, that 22 is the autologous chromosome, and one pair is the sex chromosome. They remain in the diploid form, that is the 23 pair. So, this is the picture of the chromosome, and the chromosome number, as I told in the human being, it is from 1 to 46, that is the 23 pair, 22 is the autologous, 1 is the sex chromosome. In the male it is X and Y, in female it is 2X chromosome, and 22 pairs is the autologous. These chromosomes are numbered according to their length of the chromosomes. This is the arrangement is from larger chromosome to smaller chromosome. So, this is the one picture of the X chromosome, and X chromosome is larger and Y chromosome is the smaller. So, you see that is one information I want to share. This X chromosome is the larger chromosome, the female contain 2 X chromosome. So, and male contain the X and Y chromosome. So, the sex determination, usually the misconception in our society, that you might have heard that we usually our society, that is a wrong concept, we blame female to, they direct the which sex baby will be there. It is not the true, because female is contributing always X chromosome, because female contain 2 X chromosome. So, always haploid, because you know in the meiosis division, the haploid in the gametes, it is haploid chromosome. So, female contain X chromosome always. So, always she will contribute the X chromosome, but the male contain X and Y. So, the baby sex will be determined, it depends on the contribution of the male. So, male, if it contribute X, the baby will be female baby. If it will contribute Y chromosome, it will be female baby. So, that is the usual our molecular part, that knowledge we should have, that female always contribute X and male sometime X and Y, accordingly the sex of the baby is there. So, there should not be misconcept. The chemical constituents of the inheritance, as I told, that is the DNA or RNA, because in a human it is always a DNA, and some microorganism or the prokaryotes, that is the virus are some genetic materials are the DNA, some are RNA, and the histone I already told. So, this histone protein, the basic protein. So, the histone protein, that H1, H2A, 2B, 3 and 4, 2A and 2B is the lysine rich protein, and B, that is the 3 and 4 is the arginine rich protein. The structure of chromosome I already told you. So, that this chromosome always in the 2 pair it remain, and there one will be the centromeres in the center point, and then the short arm is the P arm, long arm is the Q arm, one will be the telomere, is the end of the chromosome. So, this chromosome is consist of one basic nucleosome and then chromatin, then the chromosome. So, it has got a centromere in the center, P arm and Q arm, and telomering end is the last strand. The another point, some chromosome it is seen this secondary constriction. Secondary constriction is another constriction which is usually seen in a telomeric end. There is a that above that is known as satellite chromosome, satellite zone, SAT chromosome. So, this satellite chromosome, some chromosome in as for example, in the human it is 13, 14, 15, 21 and 22. This chromosome usually they contain the secondary centromere, the constriction, secondary constriction and one satellite zone is there. That secondary constriction I told, this is the picture that in the centromere at the telomeric end another constriction point you will find, and to distal to this secondary constriction, this is known as the satellite zone. So, the as I told, these numbers usually seen in the human it is 13, 14, 15 and 21 and 22 are the example of SAT chromosome. They are known as SAT chromosome. Structure of the SAT chromosome I already told that there will be a centromeres, that is the and secondary constriction, that is very important, and distal to the secondary constriction is the satellite zone. So, telomere in another is a important part, that is the end of the chromosome. In a human being, because we see that chromosome, why we read actually, why we have to know about the genome and the chromosome, the basic structure of the chromosome, because in the basic classes I want to emphasize you all, this chromosome structure is helpful to know or to diagnose some diseases also, because so many abnormality. If you see in the chromosome, as per commonly I will give an example as the translocation in the from 9 to 22, the translocation, that is see in the chronic myeloid leukemia. That means, I want to emphasize you these structural knowledge will help to diagnose some cancerous condition also, some abnormal condition also, that abnormal chromosome, if that another condition I want to give you example, that the repeated aberration, in the repeated aberration cases we usually examine the chromosomal abnormality. Mean, you see, nature is very clever, nature is very important, that they do not want any abnormal fetus has to or abnormal baby to be there. So, if there is a gross abnormality, mean there is a defect in the chromosome, nature will abort out. So, repeated aberrations occur. So, the product of concept is examined for the chromosomal abnormality. This telomere has also a great importance, that the telomere in the repeated cell cycle, the cell division, the length of the telomere is reduced. So, which is controlled by the telomerase, an enzyme. So, this study of the telomerase also help or it acts as a marker to know some cancerous growth and the cell division or the age of the cell also. So, these are very important things. These we can, if you know these things details, so it can help to diagnose so many diseases. Structure, this chromatin, chromosome and chromatid, this is the information. One this nucleosome, each nucleosome units in the DNA known as chromatin, then back to chromosome and the structure whole known as chromosome. This chromatin, if you stain this chromosome, the two type of, that is basically euchromatin and heterochromatin structure, basically two, hai na. So, this euchromatin and heterochromatin, this if the chromosome is active for a cell that division phase, so there what happened, that staining is very faint and there will be light, and if it will be very inactive, a non-active zone, there will be packaging will be very tight and the dense color will be there. Like this in the picture, you will see euchromatin is the lighter zone and the darker zone is the heterochromatin zone, means it is a inactive zone. These are usually that if you see in the DNA, it is the AT and GC base pairing. So, usually euchromatic zones are the usually GC rich regions and the heterochromatin regions are the AT rich region. This within the Giemsa stains it is seen. Solenoid model, already I told you that 2 nanometer structure, when this chromosome is the 10 nanometer, then is packed to 30 nanometer. This 30 nanometer again is coiled and the packed, which is as a solenoid form. So, this is a solenoid model, the Kornberg and Thomas give in 1974 this model. This is a pictorial form what I told. This one nucleosome, you see how this octamer histone protein is there in the periphery, one knot is there which is H1 stabilized by H1. This is a basic unit. The DNA, nucleosome, that is the nucleosome, then chromatin and the whole chromosome. The 2 nanometer, 10 nanometer, 30 nanometer, 300 and 700, the 14 nanometer structure how it is packaged. This is a diagrammatic representation for your better understanding. Type of chromosomes, usually this is the homologous chromosome and the heterologous chromosome. Both both is the same, it is the homologous. If it is different, that is usually in the sex chromosome, and the both types are different. So, sometimes it is the heterologous. Function of chromosome, it is the storehouse of the genetic material, that is the inheritance, that is the main function of the chromosome. The distribution, they ensure the precise distribution DNA to the daughter, inheritance. This chromonemata is the again the structural, the spirally coiled central filaments of the chromatid carrying all the inherited instructions, that is the genomic instruction. Classification according to their centromere, they are metacentric, subcentric, subcentric and acrocentric. Acrocentric means where the centromere is there, because there is no p arm, and when the centromere and the center, it is metacentric, and when it is deviated little bit from the center, it is the subcentric. These are the according to position of the centromeres. So, according to total length of the chromosome, they are divided from or grouped to A to G. A 1, 2, 3 is the A, B is the 4 and 5, C is the 6 to 12, D is the 13, 14, 15, E is the 16, 17, 18, F is the 19 and 20, G is the 21 and 22, and X is the sex chromosome, that is X or Y. In the male it will be X and Y, and in the female it is the XX. According to length of the chromosome, they are grouped usually. This is general classification. Or according to number of the centromeres, it is the monocentric, usually it is monocentric, sometimes it is dicentric, polycentric and the acentric also. All the 46 chromosomes are monocentric. In human being, it is monocentric, and different other also the centromeres, according to the number of centromere also they are grouped. Nucleus organization, that all these DNA are packed inside the nucleus. This organization pattern which we are studying now, the chromosome, that is they are in the nucleus. Chromosome number I already told you, that is from 1 to 40, that 22 pairs autosome and 1 pair is the sex chromosome. They are all diploid in number. In the all the somatic chromosome, they are 2n number, that is the double in number, they remain in the pair, and in the gametes, the sperm or ovum, it is the haploid numbers remain. In the male it is X and the female it is XY. Size, that I told in the 1 to they are grouped according to the size. The larger one, 1, 2, 3, 4 like this, this ideogram or the number of chromosome arranged according to the size, shape, they are arranged. So, these chromosomes are of larger size, smaller size, like this, they are according to size they are arranged. Karyotype is the study of the chromosome, that is anything you know details about their structure, their function and the variation. This abnormality, because why you will read something or why will you know something. So, you want to know details about it in any abnormality that you also know. So, karyotype is the usually the to study the general morphology in the somatic and the chromosome, somatic chromosome or also sex chromosome. Usually this is it needs a specialized laboratory to prepare a chromosome, but it is not that very hi-fi laboratory is there, but the basic is a good compound microscope is there and your basic cell culture laboratory is there. So, karyotyping, to know it is a art, when you will prepare very gradually practicing. So, you can do a very good the ideogram. So, the karyotyping, for a karyotype you need a basic cell culture laboratory. So, usually for example, in human being we usually take the 5 ml of blood, then that phytohemoglobin is treated and with a that is the cell culture medium, and phytohemoglobin is a plant extract and it is a cell stimulant and that cell cycle stimulant. So, in the cell culture for the 20, that is the 48 to 72, usually 72 hours, after that the colchicine is added to metaphase arrest. So, that the chromosome in the metaphase is highly coiled and organized. So, it is easily visualized with the staining. The staining usually Giemsa stain, the common Giemsa stain with the trypsin, because this DNA is coiled in a protein. So, the deproteinization has to be done for this staining. So, trypsin is the protein digester. So, and some hypotonic solution if you will put, the cell will imbibe this hypotonic solution and that it will become more fragile. So, when you will put a drop on a slide and fix it that and stain it with a Giemsa, you will see the chromosomal spread you will see and accordingly arranged. So, this is the schematic diagram from taking the sample to the chromosomal spread. This is the usual, the more precise form, chromosomal spread, and accordingly this group A to G grouping, there is 1 to the larger size are, you see according to size they are arranged in the ideogram. These are the ideogram karyotypes. You see, this is the ideogram. So, this is the length and the bending pattern, you can diagnose the abnormalities. There are so many conditions are diagnosed, the genetic condition, chromosomal abnormality, the leukemic condition and other, the Klinefelter syndrome, Turner syndrome, commonly you might have heard the Down syndrome. They are all these chromosomal abnormality, it will easily diagnose. Nowadays, in the modern the molecular karyotyping, this FISH method, that fluorescent in situ hybridization FISH pattern, it is now helpful, very much helpful to diagnose the so many the cancerous, usually the leukemic condition, the blood cancer condition. So, it helps in proper diagnosis of the leukemia condition, the blood cancer condition, and also it helps in the prognosis, because after treatment the patients, when treatment is given, whether the patient is responding treatment or poorly responding or very good responding and totally non-responding, you can also segregate in the chromosomal pattern. And also you see in the Down syndrome, you see now these karyotypes help this to diagnose the Down syndrome. So, the Down syndrome is very common nowadays, because it usually is seen in the late pregnancy. Late pregnancy mean if a female carry or pregnancy is there in above 35 years, that is the usual statistic. It is not that every 35 years above the lady will have a Down syndrome baby. It is not true, but statistically it is seen. So, nowadays the couples are planning their family little bit late. So, usually it is practicing that the Down syndrome has to be screened out, and these are the things, this karyogram or ideogram helps in the diagnosis. In the as chronic myeloid leukemia, there is translocation from 9 to 22, it is seen. This karyogram from the bending pattern and the because this chromosome the bands is fixed in a fixed chromosome. So, if you any alteration you can diagnose. G bending, this is the Gimsa bending, the usual common bending, it is Gimsa bending. Application of karyotype, I told that it is a help to detect the chromosomal abnormality and identification, loss and addition of the chromosome, and it determine the risk of the individual also, whether the individually prone for different chromosomal abnormality. This also can be diagnosed, and also in the cancer diagnosis also very important in the, it helps in the karyotyping. And the karyotyping, as I told, the CML is also, this is a chronic myeloid leukemia is very important, and other so many cancerous condition also by the molecular karyotyping, that is the FISH method, fluorescent in situ hybridization technique also it helps. See, alteration of the chromosomal structure I already told. These are the another point is the bar body, you might have heard, sometimes you should know the bar body, this is the, it is related to X chromosome. In the normal female it contained 1, this type of bar body you can, it also help in determination, it is the non-functional X chromosome. Chromosomal study helps us basically to know the normal pattern, and abnormality help in the different diagnosis, and the chromosomal pattern also help in the prognosis of different diseases. Thank you very much.